Hi everyone, I'm Victoria Blackbeard, consultant at Aerodyme, and in this exciting episode of our Diamond Insights TV Kite miniseries, I speak to Rohin Iyer, who, as the Global Program Manager for Cell and Gene Therapies at Cytiva, is literally at the forefront of commercializing these potentially curative and life-saving cancer therapies. My top takeaways from our interview is that when it comes to CAR-T, the product is the process and vice versa, which means that even the smallest tweaks in process operating conditions can be responsible for the commercial success of a product and really separate it from competitors within the field. Secondly, once a process is actually established, it becomes very difficult to incorporate next-gen technologies into it, which can actually see emerging biotechs leapfrog established players. And lastly, while there's been a lot of innovation within the space, we can expect a lot more to come, especially around workflow simplification. So these are some of the topics that I cover in my interview. I hope you enjoyed the episode.
Thank you for having me. It's a pleasure to be invited to this topic, and it's nice to see you. So I hope everyone's well in the UK, and certainly things here in Canada are improving. But yeah, it's an important topic, and I'm glad to speak about it.
Great. So Rowan, here at Aerodyne, when we talk about CAR-T, we always, use the mantra that the product is the process. But what I'd like you to do is perhaps just start off outlining for our viewers why the manufacturing process of autologous cell therapies really is so complex and so much more convoluted than what we see in the sort of small molecule biologic setting, and why that, effectively results in these products that are costing between $300,000, $450,000 per treatment.
Well, firstly, it's an important question, Victoria, and I think the unfortunate part of it is that it has a not so simple answer. When you think about how complex it is, it's related to the number of unit ops that are needed to produce CAR-T therapies to a large degree. If you think about just the viral vector manufacturing portion of the actual process, which is just a single unit op in a CAR-T process, but it's actually its own entire process to produce the viral vector alone. That alone adds, up to 50% of the cost of the process right there. So you were talking about, the cost of the product. Think about the reagents that go into it, just the reagents alone. Then, if you add on top of that the layer of these are therapies that have curative potential. So they're living biological factories. They're not small molecules that are chemical in nature, or even monoclonal antibodies that act for a limited duration but eventually lose their potency in vivo. These are products that in some cases have been detected in patients for weeks and months and years after the patients have been treated. So that really illustrates the sort of biological nature of not just the product, but the process that goes into making it. So you really have to treat them as the cells in your body would be treated. They have to be maintained under biologically, physiological conditions, relevant conditions that, keep them alive and healthy. And even despite that, many processes have built-in fail-safes to avoid losses along the way. But, when you think about all the unit ops, there's the initial isolation, the activation, magnetic separation, the transduction, viral transduction or non-viral transduction, the expansion that comes after that, the cryopreservation, sometimes multiple cryopreservation steps. You could lose, 50% of your cells just in one of those steps, leave alone along the entire process. For example, cryopreservation, it's been reported you can lose 40 to 60% of the cells just in cryopreservation. So, it is a complex balancing act, but I think the reward at the end is that you have a curative therapy. And if you can really design a robust sort of process that is able to mitigate all of those potential pitfalls, they're going to exert their effects in vivo for days, months, years in some cases. So, we really need to put our best effort into them and really try and make sure that they get to the patients as quickly as possible without suffering the ill effects of the manufacturing process.
Right, but it really is this, highly complex, customized, personalized process every time that cells are taken from a particular patient, they're genetically modified and then reinfused back into the patients. And that process is sort of, universally agreed upon and known. But when it comes to the actual specifics that every pharmaceutical company is applying, we know that there's a high degree of variation. But those variations are also the industry's kind of best kept secrets. So can you maybe talk to that a little bit, the sort of competitive edge that these slight modifications in the manufacturing process can give?
Well, it's like you said, the secret sauce. It kind of goes back to the product being the process, the mantra that many of us have adopted or believe in. When you think about it, the nuances of that process are really what make that process unique. So for example, a cell population that is harvested may be a subpopulation of a larger population of a blood cell population, for example. When you think about how you isolate such a specific population, that starts to get into the realm of, what IP can we develop around this? How do we protect this process? How do we protect this product, right? That is the process. And then the CAR construct that's used to modify the product, right? So many people are now using either viral or non-viral vectors, but what are the sort of specific features of that CAR construct, right? Does it have a payload delivery aspect to it? Does it have a kill switch on it? Which T-cell receptor domain have they used? What are the sort of co-signaling domains of that CAR construct? And then when you think about the equipment that's used, right? There's specialized equipment used at every point along the process. There's bioreactor conditions that may be very proprietary in terms of the agitation rates, the speed at which your oxygen is delivered, the rate at which your oxygen is delivered to the bioreactor, the volumes that you're perfusing every day. These are all very specific to each product, and making them in an industrially relevant manner is what makes that sort of the secret sauce, so to speak. So when you add it all up, it's no wonder that people consider the product a process and why they try to protect it. It also represents years and years of innovation in R&D. When a company like ours, like Cytiva, develops these bioreactors, and then companies go and use them in their process. So you want to protect that sort of IP around the equipment, the process, the CAR construct, the biology, and all of the work that went into that really needs to be protected so that you can monetize it appropriately.
So thank you so much, Rohin. I think, what you're saying is absolutely true. And I suppose when we look at Novartis, for example, who really struggled to initially meet their release specifications for Kymriah. And then earlier this year, we also saw that BMS, because of a manufacturing sort of hiccup, actually missed out on getting FDA approval for their CAR-T Liso-cel before the end of 2020. And I think that had repercussions of about 6.4 billion in what should have gone to investors effectively going down the drain. So what are your thoughts around how maybe easy or difficult it is for a company to be able to reliably produce these CAR-Ts consistently by themselves or with a manufacturing partner?
Yeah, it's not easy to do that, especially when we understand so little about how they work. We know that they work, but sometimes we don't know the mechanisms behind these things. And we have to keep in mind that with every patient, every autologous CAR-T patient has a different product, right? Because this is coming from an individual and going back into an individual. And so these are patient-specific therapies, right? So there are bound to be some patients who just don't have good starting material because they're already very compromised. Perhaps they're in an elderly patient population. But developing robustness into your product is really the key to success, I think. And this is why you really want to look into, you mentioned the idea of working with a development partner. A CDMO often has worked through all of these challenges. They have experience in doing this. And Cytiva has an aspect of its business called fast track services that offer CDMO services. And these manufacturing failures are not often just the biology that are causing them. They can often be just that, an operator worked on the process and another operator worked on it a little bit later, and then it resulted in a change that caused some variability. So it can be operator to operator variability. It could be equipment failures. It could be manufacturing failures. So an expert that has already worked through all of that is really going to be able to tell you, here's where you want to focus. Here's the kinds of things that could go wrong. Here's how we're going to address that to make sure that you have a robust process that doesn't suffer from these sorts of manufacturing failures down the road. And ultimately, this is what we need so that our patients are not going to be impacted down the road, because it's all about the patients at the end of the day.
Yeah, absolutely. Couldn't agree more. And we obviously, you just touched on it, there could be a lot of variation within these processes, and we know that the next generation of these kind of technologies is going to try and almost auto-correct for these and, I suppose eliminates a lot of that variability that we see. But we also know that once a process is established and, there's a CAR-T on the market using that process, it's very difficult to use comparability studies to actually, improve it and incorporate these automated sort of self-correcting newer technologies. So how easy or difficult is it going to be for almost established pharma companies to incorporate them? And is there an opportunity for maybe, emerging biotechs to leapfrog commercial CAR-T producers with their current sort of processes?
Yeah, I mean, it's a fair point. Some of the larger pharma companies that have already received approvals, they're in an interesting position right now, if you think about it, because their products were approved, several years ago now in some cases, and that has created this. Well, I guess it's a good thing, in some ways, because they've got an approved product and they've set the precedent, and they were the first to market. But it also means that they're kind of locked into a process that may or may not be the most, the latest and the greatest, so to speak. And so for them, the innovation that you're referring to is really going to happen not within their current process, which is sort of locked in from a regulatory standpoint, but more what's their next generation process. But like you said, the leapfrogging effect of some of the smaller to medium, the emerging biotech sector, or quote, is where a lot of the innovation actually is happening right now, because they're still in the R&D to preclinical stage. They have an opportunity to design in some of the coolest equipment, perhaps the stuff that is not conventional, but is now really taking hold in some of the latest CAR-T products and processes. A good example of this is, Cytiva recently acquired Vanrx, I'm not sure how it's pronounced, but it's an aseptic filling solution that we've acquired into our offerings. And, that combined with the CAR-T manufacturing flexible solution that I mentioned earlier, it really allows you to sort of close your process. It allows you to do the things that you need to do to automate it and enable better scheduling of patients. And there are other offerings out there, of course. Another example of that would be the one box approach, where rather than the modular unit ops, you have a single unit op that kind of takes care of everything. And so those innovations are really starting to become important. It'll be an interesting thing to see which one kind of crosses the finish line, which one really stands the test of time. I don't know which one will, obviously, but I think that there's enough innovation out there that people will start to realize that this works and this doesn't, and we should really focus our efforts on the thing that does work.
Yeah, absolutely. I think with so many of things, the proof is really in the pudding, right? Can that, process that is using all these next gen technologies actually produce this consistent and reliable product that can go on to, achieve regulatory approvals? I suppose the next sort of question that I have for you is, it's an exciting time for the industry too, with the imminent launch of sort of a wave of BCMA targeting CAR-Ts. We know that multiple myeloma is much larger patient population than we've seen in the lymphoma sort of setting. And that kind of step change in patient population means that we're going to see a sort of step change or increase in demand from a processing side to be able to deal with this new population that will become eligible for CAR-T products. How is that going to, sort of impact where we see the technologies and the processes develop, sort of from your perspective?
It's true that, we're going to see a lot of CAR-T products are starting to come to the forefront. A good example of this is the BCMA indication. But at the same time, I think that we still have a long way to go. So make no mistake about it, we've just barely scratched the surface of the sorts of challenges ahead of us in CAR-T. And I think the biggest change that we can expect to see will be sort of a greater focus on workflow simplification. For example, how do we reduce the operator time? How do we, reduce the number of touch points that we need to be able to, sort of do the process? So a good example of this would be sort of the VIA capsule, right? So Cytiva has recently put out a new product called the VIA capsule, which is really a liquid nitrogen-free shipping system, but it's got a lot of other features built into it. It's got integrated smart monitoring. It's got Chronicle software, which is sort of like your logistics of your logistics on your e-sign-off and electronic batch records and all that built into it. And that sort of reduces your vein-to-vein logistics, right? Because you're not having to deal with paper batch records. You're not having to deal with refilling your liquid nitrogen-based cryoshipper and things like that along the way. So I think that's a big push. It's the reduced brain domain logistics and workflow simplification. What we're also going to see, I think, is trying to reduce the expansion time frames, right? We've already seen that. The initial attempts at T-cell manufacturing with rapid expansion protocols used to take 14 days, 21 days, and that sort of thing. What we're now seeing is an 8-day process, and we're starting to see a reduction of that 8-day process too. And I think there's also going to be a push toward allogeneic strategies. I know we were sort of focusing on autologous CAR T in this day and age, but there's a lot of activity and momentum in the allogeneic space as well. And what we're going to see is some efficiencies, I think, in terms of borrowing some of the learnings and lessons from the bioprocess industry, where we've already been manufacturing products that, like vaccines and mAbs, for example, using huge bioreactors. And these are lessons that we can directly apply in some cases to production of allogeneic cell therapies. So I guess unlike the autologous approach, we're going to start to see maybe a little more innovation at a larger scale, and that scalability is going to allow us to treat more patients. Like I said, there's a lot more work to be done in terms of safety and efficacy, both in the autologous and allogeneic spaces. So it'll be interesting to see what sort of wins in the end. I think autologous CAR T therapies, however, are here to stay for the foreseeable future, just because of how well they're doing and the fact that they're addressing a really key demand right now.
Yeah, absolutely. Really exciting times ahead of us. But, Rohin, that is all from my side for today. So thank you so much again for, giving us your time. This has been a fantastic conclusion to our sort of CAR T mini-series.